Ta-da! I've now got a ruler. Hopefully you always have a ruler with you. This is an experiment into the enzyme catalase. The student homogenized. So homogenized is basically bashed up and mixed, turned into kind of mashed potato. Homogenize, homo means it's the same consistency all the way through. And they probably did that in a pestle and mortar, and put the water in, put the potato in, and mashed it up. And then they had little discs of paper, 5mm in diameter, so about that size, and they've dipped them in the potato extract. So they've dipped those in there. And then they push it to the bottom of a test tube. So the test tube they have got there, they fill this test tube with hydrogen peroxide solution, and then they put the disc in, put it to the bottom, and then over time we get the reaction producing bubbles of oxygen, and then this disc will rise. And they've timed how long it takes for this filter paper to rise to the surface with the bubbles of hydrogen peroxide on. Using the same hydrogen peroxide solution, they've told you that for a reason probably. The experiment was repeated twice to obtain 3 results for a range, and there's the standard looking table. Calculate the mean rate of reaction for the 0.75% hydrogen peroxide solution. So that's going to go in there. So rate of reaction, so if we're doing the rate, it's 1 over the time taken. So the rate equals 1 divided by the time taken. So trusty calculator, I can't use. So we've got 21.3. 1 divided by 21.3, and that is 0.0469. But we've got 10 to the 2 here, so that's 100-fold. So they've times it by 100 to make the numbers look nice. So we times it by 100, and that's 4.69. And now we've got 4.69, but the table has got 1 decimal place, so we're going to go along with what the style of the table. So I'm going to put 4.7 there. So 3 things to do in that calculation. The rate is the 1 over the time taken. Then we are timesing it by 100 to account, because that's what they've done to make the numbers look nice. And we've used the same level of precision as given by the numbers already in the table. Plot the data on the graph below. And there's 3 marks for this. So the marks for these are the axes are correctly assigned with the correct labels, so putting the correct labels on the axes the right way around. The scales and the units for the axes are another mark, and then there's 1 mark for plotting the points and drawing the line on. Plot the data. Which data? Well, we've just been asked to calculate the mean rate, so I'm going to plot the mean rate. So my independent variable is my concentration of hydrogen peroxide. My dependent variable is my mean rate, and that's what I'm going to plot. My independent variable goes on the bottom. So my independent variable goes on the bottom. And I'm just going to copy that out like it is there, and then I can't get it wrong. So it's concentration of hydrogen peroxide solution, and the units are percentage of stock solution. So why wouldn't you just write that out and copy the units? It's got to be right, hasn't it? And up the side, the same thing. Mean rate of reaction, and the units are seconds minus 1, 10 to the 2. So per second times by 10 to the 2. Scales, so we need to go up to 6 here. They've provided a rather handy sized bit of graph paper. So 1, 2, 3, 4, 5, 6, it just fits. So 0, 1, 2, 3, 4, 5, 6. By the way, use a pencil for this graph. I'm just doing it in ink so it stands out in the video, but you'll use a pencil so that you can rub out errors. On videos I usually make an error at some point, so let's see. Mean rate of reaction, that goes up to 9. So we could, where could we start this? We could start this maybe 4, 5, 6, 7, 8, 9. That could do. We don't need to start this at 0, so I'm going to start mine at 4. And then 5, 6, 7, 8, 9. And then I think I'm using more than half the available graph paper. So let's plot some points. So 0.75, so there's, each one of these is 0.1, so 0.75 is about there, and 4.7, 4.7, so that's the hardest point to plot really, that one. 1.5, 6.5, so 1.5 and 6.5, 3 and 7.9, 3 and 7.9, that's easier to plot. 4.5 and 8.5, so 4.5 and 8.5, count those halfway, and 6 and 8.5. And in biology, most of the time, we can join dot to dot with the ruler. You could draw a freehand curve there, but you'll always get the marks drawing a nice line, dot to dot with the ruler. Try and get it to the center of every point. There we go. We can't extend the data to 0 or 4 or whatever because we haven't got any data there. So that's the only line that we can draw. So 3 marks, 1 for the axes, the right around, and you're just copying that, so you can get a mark for that. 1 mark for choosing the scales, and 1 mark for the line and the points. And then it's wanting us to explain the shape of the graph. So we need to explain why this is going up and then why it's leveling off. We've got 6 marks available here, so we need to use the right terminology and get the right details there. So between 0.75 and say 4.5, where it's going, we're explaining why it's going up. So between 0.75% and 4.5% hydrogen peroxide. We don't need to say it goes up because that's describing it, and we're just trying to explain it now. As the concentration increases, the rate of reaction increases, and that's why that time is taking less. As the concentration increases, the rate of reaction increases. So that's really our first mark. Why? Because there's more hydrogen peroxide there, there's more enzyme substrate complexes formed. So as concentration increases, there are more enzyme substrate complexes formed. You could also say there's more chance of a collision between the substrate and the enzyme, so more chances of collisions, per unit time. And we could say that the thing that's stopping this from increasing anymore is this concentration of hydrogen peroxide. So hydrogen peroxide is the limiting factor. So we've explained this part of the graph, why it's going up. We need to explain why it's leveled off. So above 4.5% hydrogen peroxide, as the concentration increases, there's no further increase in rate. As the concentration increases, there is no further increase in rate. The reason for this is all of the active sites are occupied, or the catalase is saturated. So all the enzyme active sites are occupied. It's working as fast as it can. And now we need to go, what's the limiting factor now? So the limiting factor is not the concentration of our substrate, it's the concentration of our enzyme. So the concentration of enzyme, which is a catalase, is now limiting, or is the limiting factor. So 6 marks, and you see we're having to bang in the marks in the space available. We can't waffle on for the first mark and so on. We need 6 kind of separate bits of knowledge. And part C, another 4 marks. Wow. Apart from human error, so not human error, that's just 2 sources of error that could lead to inaccuracies, and 2 ways in which these could be minimised. So we're going to have to give an inaccuracy and the reason how you can minimise it, and then another inaccuracy and a reason and the way that you can minimise it, and that's going to be your 4 marks there. So you're going to say 4 pieces of information. Apart from human error, so we can't say we couldn't use a timer very well, or that sort of thing, or I forgot to put the solution in the tube. Not that it's that kind of errors that are built into the experiment. The first one that I would think of is the bit it gave us right at the beginning. It said using the same hydrogen peroxide solution. So they've done these experiments. When this one's gone, they've put another disc in the same solution. That's risen to the surface. Then just put another one in. But you're using up the substrate as you're going along. So eventually, if you just kept adding more discs to this, it wouldn't work any longer. So that's my first one that they've given. So used the same hydrogen peroxide solution each time. Now the concentration would decrease, and so the rate of reaction would also decrease. Our solution to that is to add fresh hydrogen peroxide for each repeat. So use fresh hydrogen peroxide for each repeat. And I mean, there are a number of things you can say here. You know, the amount of time you put the discs into the potato mush, it didn't tell us how long those were, so some might have been for longer than others. But I think you'd be better off with some of the general suggestions that you can use in almost any experiment. Always look for, have they controlled variables such as temperature, pH, volumes and things like that. That's what we usually go for. And as this is an enzyme experiment, we go, what affects the rate of enzymes? Well, temperature and pH are kind of obvious ones. So I'm going to, the temperature wasn't controlled. The temperature was not controlled. And this is actually an exothermic reaction, but it could be endothermic. You know, it doesn't actually tell us, but it doesn't really matter. The temperature was not controlled, so use an thermostatic water bath to control the temperature. And that's such a common suggestion you can use. And if the experiment's about temperature, you could suggest the other one, pH. So you could, the pH wasn't controlled, and so use a buffer. Another easy one. We could say other ones in this one that you might not have thought of. When you mush up, it says it produced a homogenized potato tissue. Well, some bits of it might have had more potato enzyme in than others. So you could make a better, use a solution for that. But I think these are the more obvious ones. So quite a chunky question, that 16 marks.